What is that? Neonatal sepsis. That baby born, within a day or two, it got a sepsis in the heart and different parts and died. And because this death, neonatal sepsis death has enhanced 30% across India's, the statistics I'm telling you now, that's about 30% death due to neonatal sepsis across. Moreover, what is happening because of COVID, even 30% COVID-related death happens, and secondary infection. But COVID is cured, but still another agents become registered, and so people are going for secondary infections. Now, this is what is antimicrobial resistance in short. Cells, microorganized bacteria, it could be virus, parasite, or fungi. And example of AMR like E. coli, which is a gram-negative E. coli, and Klebsiella or Acinetobacter, so many things are there, which called getting resistant and all those. And causes of antimicrobial resistance is one of these things what has enhanced much more, because in the COVID time, because we don't know what medicine has to be taken and all, in order to save the life, doctors has prescribed a lot of medicine. And that medicine, I'll come into that in the real data, whether it is effective or not, and what happens because of this overuse of different kind of chemical reagents or drugs during COVID time. Another very, very serious question is called OTC, over-the-counter availability of antibiotics. And that is, there is some rules and regulations are there that without prescription, you should not take antibiotic, but it is not happening. And now recent government is taking measures, I'll come into that. But one is the very important part, what I'm just going to tell you, all those who has participated here, it's a very, very alarming fact. 75% of total antibiotic used in farming. This is called either this is cultivation, animal farming, or cultivable for other things. 75% of the total antibiotic used in the world, it is go for farming. And those antibiotics, what are those? How they come into circulation and what they are doing it, that is the major concern throughout the world we are thinking and all. And now, another thing I just want to tell you, this is colistin resistance E. coli. It is coming, colistin, that is a kind of things. And this E. coli resistance to colistin, it is from farming and it is coming to the circulation and widely doing a lot of kind of a disease for the people and all. And this is the super bug called Candida aureus. Why it is so dangerous? Because there are two kind of infection is coming into one called community infection, when going through the environment where you are staying. Another called hospital mediated infection, and that is much more dangerous. You might have seen everybody. Doctors do not want to keep patient in ICU, CCU, if it is not required for a more time, because many infection which is could not be treated is coming from this ICU from the hospital. Here also we are participating in different meeting, Royal Society of Chemistry, Royal Society of Biology, Royal Society of Medicine, or several international doctors forum and all. What they are thinking that ICU practice of the hospital or IV injection, the schedule or what parameter needs to be changed throughout the world. So new rules will come and we have to learn from the doctors and we have to talk to them and we have to adhere to this rule. Most important, this Candida aureus, even you try to wipe it out, it stays in hospital. You see this, I put that commonly positive sites, where is this ICU and all those things. There this Candida aureus can stay up to 1 month. It has its own life and things, it's much more difficult. And you see that in those hospitals, those who are all those that if things are coming after washing, if that is exposed to the environment without properly treated, that will be circulated and coming back to this very quickly. So this is a major concern, what I just want to tell you. Now, challenges are how to do it, because, India and China, more or less 40% population of the world. And so here I put in mortality rates in African Asia, but it is also, other things are also there. And economic burden will be 2001 trillion. But will be coming a main point. Exactly what happened when we need to take care, we are not taking care of. But when these things is out of control, we are throwing our money or everything to do that. That time we can't come back. So here is the discussion. I'm giving some kind of a scientific data or clue. And for all audience who joined off, they're scientists, they're general people and all, but we have to learn from them. And when discussion will happen, we have to talk and we have to finalize. So not only awareness, we need to practice. We need to invent something through that. So now this is the initiative has been taken, different declaration, both Indian and international, and way forward is putting more and more money for new drug discovery or these biopesticides or fungicides, and all those things need to be adhered to. So, but in another 30 minutes discussion, I'll not go for all those points, but I'm just doing it. This I got the question from this different time for a couple of years from the audience. That's why I put together. But in a lecture, I'll mainly focusing on the very, very basic thing. And I also want to learn from you what needs to be done and what could be done from that side. If you have any question, you can stop me, or I can do it, or end of the thing, you can take it. So now the question is, this is like a circular 360-degree things. Here is a slide, you can tell, you can see that antibacterial resistance from the farm to the table. It's because if it is the antibiotic given to the animals, then it can go that kills some bacteria, some will be resistant, that will come to the spread through the different foods, and then it will go to the animal. When animal poops, again it will come to the contaminated food or through inhalation, and then ultimately this impact will be happening. So the question is very simply, what I just want to tell you, resistance, spread, exposure, and impact. Now coming to a little specific about this. What antibiotics are used for crop disease? It's streptomycin, over-the-counter, several antibiotic used for the production of this crop and this animal farming. But the most danger is, here also we need to think of these greedy people. Here, if you use the exact dose of antibiotic to the animals or those, your yield will be less. So it was the common practice to use this dose of antibiotics less. So it can't kill the bug. It is allow bug to be resistant, through which I'll come into that. So what are the antibiotics used in animal farm in United States? 80% sold used in animal agriculture, 70% are medically important. And antibiotics can increase animal performance. Very low dose. See, here I'm just telling, if you want to get some kind of a meat for few kilo more, low dose antibiotic they're giving, it can enhance the performance, enhance them. But in contrary, it is allowing bugs to grow. Let's see this. This is the figure of a cattle. And you see there are 4 parts. And the most important thing is the rumen parts. And this rumen is the major things which can carry all kind of infection. Now, if I tell to the human to translate this thing, if you have a 70 kg old, 70 kg weight of the average weight of a human, out of 70 kg, 2 kg will be your bacteria, which is there remaining in your guts. All are good, some are bad. When you take the antibiotic, you have to put some bacteria will be you have to supplement this would be dangerous. So this is the circulation. What I just want to tell you here, from the farm to the table. So the two major points: 75% antibiotics are using for animal farming or agriculture. Major infection, what is community acquired infection, another is hospital acquired, is much more dangerous. Now let's see this, how the antibacterial resistance happens. Actually, the primary infection, if you get inadequate antibiotics, and how it is doing it, that's why I'm just going to come and tell you. This MRS is one of the dangerous, very dangerous infection to be treated. And most probably you can tell that 2002, linezolid is the last antibiotic has been discovered. Before that, antibiotic drug discovery or coming from the new drug to the market is very less. But one good news is recently Okhad has a new antibiotic, and they are putting to treat pneumonia of a different, could be $9 billion worth. This make India and our proud, and something's also coming into the pipeline, but it is not very always encouraging. Compared to other therapeutics area, antibiotic drug discovery is not that encouraging, though we are in a crisis for antimicrobial resistance and coming. Now, understanding the antimicrobial resistance, from there I'll go very slow. Here you saw that antimicrobial resistance showcases the survival of the fittest. Principle in bacterial evolution, going phenomenon observed across all organisms. Survival of the, you have to see what happens actually. You know, there are two different things. If you have a diabetes, the drug, it should be effective. It should not be that toxic to the human cells. And on the contrary, it should not cure, but control the onslaught of the disease. And this treatment period is month, years, or lifelong for this others lifestyle management disease. But when you are get infected with antimicrobial, it's a foreign thing. You have to kill the bug. It's a completely different. You have to kill the bug. That means not control the onslaught of the bug. You have to completely kill the bug. If you have a little 5% bugs also there, that got resistance, your disease will come back. So that's why you need to take this antibiotic with the supervision of doctor's prescription. Let's see, given one simple example. Previously, azithromycin, which is the upper infection, it was 500 mg for 3 days. Now it is 6 days, going to be changed to 10 days. And that also, when doctors prescribing empty stomach or with food, completely different mode of action for azithromycin. Why it is getting resistant? People take without showing to the doctor, just take the azithromycin. My fever is over. After a couple of days, I am cured. So that means you kill maybe your 80% of the bug. Still 20% of the bugs are there, and they're very clever. They want to metabolize this drug, got to the resistance. So always you have to be put your body into selection pressure until all bugs are getting killed and you are cured. This is a major concern for antibiotic resistance going. And here I just want to tell you what happens. I'll come to the last part of my talk, how I'm telling. If you go to the village and all, ladies are getting more and more, that is called urinary tract infection. And you see that infection, when you saw the report, 10 to the power 14 is the titer value. Being infected much more and more, because the poor people, female, especially the females, they can't afford tissue papers after the toilet and all those. So once it's going, and it's a very dangerous symptom happens and happening, and the deaths are happening. So it has to be very taking care of this thing. I'll come into the last part of point. And then over the time, this has got resistance. Now the question is, what are these mechanism, or what kind of a things is getting resistance? See here, I just want to tell you, I know some scientists are working on there. They're scientists, some are from the common people, but I try to balance my talk, not going too much into details or without going my details. The reason is I also want to learn from all of the audience what we have to do. Is there any new novel idea we can do that? But just to tell you, see what happens, this bacteria cell, in case of a tuberculosis, gram-positive cells, it doesn't allow any drug to be penetrate. This methicillin-resistant Staphylococcus is a gram-positive. It's not like TB, but it's also dangerous. It didn't allow drug to go in. Then, if you have a gram-negative, double layer of bacterial things, E. coli and all, drugs to go in much more difficult. So these are the major things, that impermeability of the drug, what you are getting discovering, and it is not going through it. And second is, whatever the things it will go and bind, and that is called target, where it will go, drug will bind and cure it. That target itself gets modified by the bacteria. They're very, very clever. Or the drugs, whatever you are giving, when inside is going in there are some enzyme which is changing the drug's property. Like just I'm giving one example, during COVID time, we have to give the oxygen in prone position because it will go to the lung. Why prone position? Because the pH of the lung is acidic. So oxygen has to go there. But pH of the is acidic. If that drug goes through the acidification, that will not going to work. So in the COVID time, what happens? Several drugs which is required to create to or some kind of a liver disease or pancreas disease, that has been given for the lung for other things. So the availability, they're changing, all those has changed, and that created additional problem to these things and to the resistance. Fourth is pumping out. That means it will take and it will throw. It is called efflux. It will not allow to go to the drug and cure for all those kind of it. This is the mechanism, but is not for microbial resistance. But now there is a puzzle, and you know, some drugs got resistance very quickly, and some drugs takes time to get resistance in the same body. Why? Scientists trying to understand why certain kind of antibiotic get resistance very quickly, whereas certain kind of antibiotics, it takes to time to get the resistance. What could be the thing? So puzzle is to understand exactly how bacteria adapt their resistance. Now the question is here. Question is, why it takes longer for some bacteria to develop antibiotic resistance while other adapt very quickly? Now the finding is very, very interesting. What happens that the question is, one bacteria, how give its resistance gene or that particular to its offspring. It's kind of a toolbox. What happens? You see here, there is a mechanism called cut and paste. So DNA double helix, cut and paste. Let's see one portion which has a resistant. It has to be cut and it has to be pasted in a different color to its offspring. How it will do it? It will do through a protein called recombinase. And the protein binds to the DNA. So it's a DNA binding protein. Now the question is, just to take an example. Let's say you want to cut some with your scissors. So if the knot is very tight, then and then you can cut. But if the knot is very loose, it is just not making knot. It's just falling. It is coming, it is falling. So the scissors can't go and cut. So that means more tighten that particular enzyme binds to this DNA sequence, then the scissors will cut so quickly, and then it can do the pasting. So here is the two things, what I just only want to tell you. If you have what is the weakness of the bacteria? The bacteria exploit the weakness of the bacteria. Because in order to make this resistant, exploit the weakness.
Okay, Azilan, please mute yourself.
Is it okay, Dr. Senapati?
Yes.
Okay, thank you. There is some sounds coming.
Dr. Senapati, you please mute Edilan.
Yeah. Is it okay, Dr. Gopalakrishnan? Can I?
Continue.
Yeah, please. Okay. So now, in order to, we need to understand this. In order to develop a drug or anything, we need to understand the weakness of the bacteria. What is the weakness of the bacteria? In order to pass this, what is resistance, which can, that gene to the offspring, can you stop it? Two process. One is called cut, another called paste, totally called recombination. Can we stop the cutting? Or if it cuts, can you put the paste stopping? Can you a new drug? This has come a new mechanism after a long scientific debate, 2024 December. So what you are telling there, that uncouple the cut and paste, the recombinant mechanism, or didn't allow this particular hairpin to any protein which recombinase to cut, don't allow binds there. Then in next generation or there, it will not be passed. So this is in the field of bacteria, a new thought of given. And to be those who are from the field or you want to know, you can think through a very high or microscope or a new thing. You can take a single bacteria and you can visualize how these recombinants and all those things are happening. That's why the detailing and things, if you want interested, I can give you later. So this is about this bacteria. Now let us come, we have just come out for the COVID. Let us think how the virus, why that COVID is so, how it enters. So this is the COVID virus, and you have the spike protein and all those things, what everybody knows, against with that our vaccine and every developed and all. Now, let me tell you how a virus, it gets the entry inside a cell thing and membrane. I just want to tell you a very small story. Let's say it is your home and your house is locked and you have the key. So what we practice? We go to the home, we just hold the key lock in our hand. If you're right-handed or left-handed, then you put the key inside the groove and just give in a trigger in the right-hand direction. Then what will happen? Your lock will be open. You got inside. That's why we do. So here the most important part is called trigger. The trigger, what is I'm giving, right hand, so it's right hand clockwise. This some pressure you are giving. As a result, that lock will be open. And other way, if you give the left hand, the things other way around, anti-clockwise, it will be closed. But if you have that key inside the groove without that force, lock will not be open. So here is the things what this COVID virus has done it. I'm just coming into that point. Before that COVID coming to things, I think here we are sitting in Hyderabad, and those who are from Andhra Pradesh and Hyderabad or from some other part also, you know, before COVID comes, there is a disease came that is called chicken disease. In the poultry, chicken they're getting killed. First is very slowly. After few months, rapid. It's millions of chickens are getting killed. So this is a kind of a disease that has killed that, and those chicken meat and all those things has been there. Don't take for all those kind of things. Why all of a sudden this chicken, that was a kind of a virus was there. And I am coming into that. What is the similarity of that virus with this? When that virus infected with the chicken first, it was not that infective. You know what happens in the course of time, that virus has acquired a particular motif or gene sequence. And what is the speciality of this gene sequence? This is the gene sequence which can be cut by the blood enzyme called furin. There is plenty of furin is available in that blood, either animal blood or human blood, and that furin can cut this particular sequence. So what I just want to tell you, here you saw that this virus, this is a color and a black one, it attached to the human cells, but it can't get entry. In order to get entry, what happens? It needs a kind of a smooth pipe kind of a thing, or tunnel kind of a thing. Through that, its genetic material can go inside. And furin protein does like this. So that same furin cleavage site which happened into this chicken has been acquired by this COVID virus. And that's why this COVID-19 was so infective. And slowly, because of mutation, its infectivity rate has been totally enhanced. So this is the mystery. How see that? A small organism, a virus, a bacteria, how clever it is. It is going and it is coming and doing the evolution pathway and getting resistance and getting entry inside. And when it gets inside, it gets only its genome, either RNA or DNA, not the entire part. Because it can't survive of its own, it has to take the raw material or survival things from the human. And that's why this COVID things and happens. So two things I told. One is the weakest thing of the bacteria, how to do it. Another for the virus, how to, that's why the vaccine and everything has been discovered and all. Now, this is a very, very dangerous point why you are bringing into the COVID. All we passed through the COVID and we survived, many people died. When that first time COVID came, what was the treatment? That is called a solidarity trial, but World Health Organization. What are the criteria? There is no criteria. If you have a COVID, you got in the hospital, do you need oxygen or not? Whether is death happens or that. There is no other things, no other mechanism of drugs known. You can see here anti-malarial drug has been given, antiviral drug has been given, HIV drug has been given, even PAN40, which is, that also has been given. People given aspirin even. And most of the thing, if you see, those who have, but those who are COVID survival, they may have taken 20 to 30 different kinds of drug that time, because that time doctors are on foot. They have to do, they don't know what is the real prescription. So this is the solidarity trial, what happens. That time happens. After that, because this is an emergency, this is good. After that, what is happening? Because of that, there are several kind of a resistance are coming. Just want to tell you one thing. I have told you, don't go for the details for x-axis and y-axis. I just want to come. This is this green things. What happens? I told you two different kind of a disease. One is a lifestyle disease, another called anti-infective. Lifestyle disease means you have to, onslaught of the disease has to be come slowly. It is not cured. It is under control. But because of anti-infective disease, you have to kill the bug. So here you can see there is a called EC50, means 50%. But in case of antibacterial, you have to kill 90-95%. Even those are there, it will come back. Even a target, if it is a lifestyle disease, you can go for the 50% of this inhibition or activation. But in case of bacterial target, you have to give 90-95%. Otherwise, it will come back and do that. That makes a huge difference. I'm not going into details. I know lots of my friends from chemistry background, they joined. They might have known, and those who are our mentor, Dr. Burishu, Dr. Uma is from chemistry, all they can understand. Those are the drugs. What I just want to tell you, I just want to tell drugs has been given without any mechanism known to kill to this COVID. And COVID actually happening in the lung and other parts, later part of the heart when the infection rate. But this is the drugs. When it penetrates different tissues, its profile is completely different and the concentration is inadequate. Later on, we found those are the things which is thought of very, very effective for killing all those. It is exactly not. You saw that the blue color bar has reduced if you take EC90, which would be effective against this COVID. So now we are in a situation of crores of people throughout the world, especially in India and other parts where there, he's given this kind of a drug for a long time, and this is they are already in the exposure, and they are no use for killing for antibacterial, but they are given. But those bacteria which is there, they are very clever and they are getting resistance. So it's very, very difficult now to cure those resistance before COVID and after COVID because of this misuse of drug. In addition to that, we take antibiotic without these doctors and all those consultations, and we don't kill the bug completely. We got cured. So now let's see another example where we work. Professor Bulushu Gopalakrishnan work. This is for the TB. You know, in the TB treatment, multiple drug resistance TB or latent TB very difficult to cure. And now, even whatever the drugs it given is very difficult. Government has put a new rule of betaquilin, pitonamide, and linezolid and all, but still looking for a new kind of a things and all. And we are thinking for a new mechanism for amino acid biosynthesis inhibitor for these things. This is, I don't want to discuss much more, but what I'm just telling it, when you treat in a disease, we're treating the macro environment. But the micro environment, what we don't take care. Let's see, if you put a baby into the jungle, the baby is human, but he will adapt it with the jungle environment like an animal and all this. So if you put a drug to cure this macro environment, still that micro environment where that cells are there, that is polluted, if you don't take care of it, then if you put a new cell, it is also going to be killed. So the point here is, the question is this curing the micro and macro environment and don't allow it to be resistant and all. So this is the scenario of the India, and this is the global scenario. What are the steps are taking throughout? And India, I'm just telling one thing. Last couple of years, 7 states has been imposed and told, and they are doing it in different meeting level that don't take antibiotic without consultation with doctor. And Kerala is the first state which has done it to some extent, and there are 1,000 crores per month of antibiotic saving misuse. But the question is coming, if you're allowing not used to antibiotic, that is one. But the question is how those people will be cured. We need to make an arrangement for them, how to do it. Let's see, we don't have a new antibiotic and all. But still, can we take care of something where we can think of that we don't want to be infected? Now, this is this resistance rate and how can prevent it. Now, I'm just telling a smart approach taken by a state of US that is called Pennsylvania. Before going into the detailing of it, I just want to tell you one thing. When you got infected, if bacteria and all those in a sufficient quantity can infect yourself, you can't control everything. What are the way of infection? Through hand, either through what is called when you take breath or from other means. The first question is, can you prevent? Can we allow that unnecessary bacteria, all those things, not to be coming inside? If it doesn't come, you don't have to treat that. That means the simple question, if you don't overeat and don't do exercise, you will not gain body weight, right? So that means if you balanced it or do that, don't allow things to come, then can you cure that? This is one of the approach was taken by the Pennsylvania. This is a state. This is a Pennsylvania state. And you see the where from they start. They started from the young. Here I'm just telling, I'm very delighted that Breakthrough Science Society three churches arranged this talk. And I know the Breakthrough Science Society have a lot of learning center throughout the country, also in abroad. I'm requesting those people, I make an appeal, those who are Breakthrough members or those who are scientists, all come forward to do this thing kind of. Here you see that this lady is a doctor. And the one question was caught a cold. How? And these are all young kids. They are there. And this doctor is explaining how we catch cold. And how is even catching cold, how it is giving to infection. And what could be the. And you can see that those kids are brought by all their parents. So now what they have showing here, I'm going very slow because this is very, very things for you. If you put a kind of a blue, this kind of a, it is just mimics your E. coli infection or kind of a thing. And when you wash your hand, because of COVID, you know how to wash your hand. And always it has been told that wash your hand for 20 seconds. Let's see, just to give an example. Let's say you have a watch, we are watching the right hand or left hand. Take the watch and you have to measure the 20 seconds. I'm taking my watch. You may just count 1, 2, 3, 4, like 20. That will mimics your 20 seconds. Why 20 seconds? Because those sanitizer or soap contain some reagent. It will destroy the lipid layer of bacteria, virus or something. And the genetic material will be out. As a result, what happens? Your germ can't infect you. This is the primary things what we did, and exactly during COVID time, other than wearing mask, the uses of sanitizer has been much more effective. Now the question comes, how to wash hand, and for the kids especially, which part of these hands has to be? Just putting a sanitizer soap for 5 seconds will not work. You need to palm, finger, and other things has to be very nice. And there is a misunderstanding with COVID time. People thought the sanitizer contains 50% alcohol. So if I drink alcohol, it will go to my things and it will kill. But there are two different things. When you drink alcohol, it will dissolve your lipid layer of your own and it will give a burning and it is bad. Here with that palm, it is not a lipid layer. It was killing this bacterial lipid layer so that you will not be infected. So what beautifully this Pennsylvania has done, and I am very, very, when I saw that in international meeting on Royal Society of Chemistry and Biology, and we also discuss this point in our government several meeting, and people I'm just telling you, all we know each other and we are there. When I discuss this thing and all, so just washing hand will do it. Then some kind of a things I got, it's body bath kya hai? What is the big things to do? But we have done it in questioning. Then next, in our government meeting, I just showed an example of a safety wing and asked all those people who are there, national, international, can you make another safety wing, which is alternative and does the same thing? That's a pin drop silence. So thing is, what I just want to tell you, if this sanitizer or washing hand can give you protection for infection, what if it is small b? It has to be very, very important things to be done. And that's why this Pennsylvania has done it. You see this, after doing that, didn't stop there. They got a competition with age 4. And how much they understood, you put a drawing. Then they go for another age 5, 6 age group student. Then go for 8 age, and then go for 9, and then ultimately the 12. And if I come back from age group 4 to 12, and what they have written and what they have done it on the sketch and the wording, you saw that maturity is going. maturity is going for these things and all. So then Pennsylvania, what they have done, they told that those parents, you go back to these kids, train those kids will train other kids, and also measure what is the antibiotic uses per month of your house. And they claimed through that they have done 50% down of the antibiotic uses, and not only antibiotic uses, also it has come down the infection. And the power of the kids is enormous. Everybody likes kids and all, and those kids going and telling this is another kind of a thing. Though this reminds me of something, though I'm not telling one thing to you. When the Nirma washing powder in our childhood, we saw that the small kid putting a white dresses, that is the advertisement come, Nirma powder works. And if you go to the invention of the Nirma powder, they given those powder to 10 kids. They went to the shop and told Nirma powder works. And those go to the kids. And that's why this particular small kids advertisement come into TV. I had a collaboration with him. I heard that from that. So my point is, can we wash our hand properly after 4 hours? We know how to wash our hand. Can you use sanitizer much more now, especially for the kids and those who are in that village, especially the female are getting infected with units, come all scientific, national, international figure, we are just getting killed because they can't afford the tissue paper and all. So my appeal to the breakthrough science societies, all those there and other scientists to get some more idea. But this is one of the idea what I'm telling. You have your learning center. Can you teach all those kids and go for, with the doctor's help, do this camp and call all those parents and execute it. Though it's a very small, some think is very small thing, but very powerful thing. So that's why my appeal to all of you. And this is the thing what I just want to tell. And I want to acknowledge Professor Dhruvadyoti Mukhopadhyay, President, Professor Sumitra Banerjee, General Secretary, my mentor, Dr. Guru Gopalakrishnan, Nilesh, Professor Jha, the President of Madhya Pradesh, Subrata Gauri, Nanda Kumarji, George from Kerala, Uma, always, thanks for your kind help always, and Manovendra Spar. All of my students of Dr. Reddy's, those who joined today, Royal Society Chemistry Biology, and I took a lot of help from Medical Service Center, which is in Kolkata, and this is the name of the doctors. I'm always in touch with them and try to solve when the new antibiotic one way is coming, and try to not only awareness, just to execute this idea of the logic. Wash your hand and wash your clothes, especially when you're coming from the hospital or anywhere. And please do that so that before new antibiotic and other things are coming, or all those things are happening because of weather, climate change, bacterial replication time also happening. Instead of 60, very quickly it is going to be tighter. So we'll not allow these things to come. And please be careful when for the animal farming and all those things you are taking. And let us build up a mighty movement. And we will, as a human, all those people together, collectively, we can, through this movement, through this awareness, through the learning, and more I'll get to know when question answer, will stop this menace, antimicrobial resistance. Thank you for your present hearing.
Okay. Thank you, Dr. Misra, for your wonderful talk, which is associated in our daily life and awareness program. So I request audience for your queries or question one by one.
Hi, Parimal.
Yeah. Hello.
So there are 2-3 queries I have. So you mentioned about, so when we take antibiotics, the gut microbiota also get killed.
Mm-hmm. Yeah.
So do we know anything about the resistance that the gut microbiota develop?
Yes, so actually it will not develop in the gut. Sometimes happens those who are suffering for urinary tract infection, those gut microbacteria come into urinary tract. That time this got resistance because, especially because of sugar reabsorption and going back and all those kind of things happen. But before of antibiotic and the gut microbe getting resistance because of antibacterial taking 3-4 days is very low. So this I tell because you have to take vitamin actually when you are taking some kind of a very strong antibiotic. That will kill you. As I told, 2 kg of 70 kg of is that bacteria, and those
Yes.
bacteria, those good bacteria become bad when it comes to the urinary tract infection throughout the body. So this is one of the resistance it happens. It's see when you do the urine culture, if you see your pH is acidic or things, then doctor tell if you're diabetic, your sugar is flowing to the urine or not. Then they will see whether nitrate formation is there in the urine or not, and which kind of a crystal that you give you the level of storage and what kind of a things has come from mycobacteria, not from outside, even inside. So those are the things needs to be depends on the disease and what kind of a things you are taking all those.
My second is a comment. So, see, you talked about the COVID treatment, right? So, when in the beginning, the COVID treatment was extraordinarily harsh.
Yes.
They threw everything. They threw the kitchen sink on every patient. For example, the doctors generally prescribed ivermectin, azithromycin, favipiravir, ibuprofen, and several other, antihistamines. Everything. I mean, there is so much overload of this medication for that whatever period, 14-day period. It was horrible. So don't you think that would have actually been responsible for a lot of post-COVID effects?
Yeah, doctor. I'm just coming into that point, but here also we need to think that time absolutely unknown when that epidemic happens. So doctors thought of there is no mechanism, nothing known. Those drugs, whatever you given, I also showed to my EC90 and EC50 analysis. Those are given somebody is for viral, somebody for HIV or something else. And as the COVID virus is RNA virus.
I forgot to mention they were also prescribing steroids.
Yes, steroids. I'm just coming to that point because of inflammation and all those things kind of a thing. So there is nothing known, and it was too harsh. You are absolutely correct. When the time passes from this COVID phase 1 to phase 2, when again mutation happens and infectivity is very high, and that time also you saw that it went beyond control by this time. So it has take time to know which is the things has to be given. Yes, it has been, but as I told you, as the solidity trial recommended by World Health Organization, for the scientist or doctor, nothing is known. So it is just use and try. It's really what you are telling correct. But that time, to saving some life, to some way it was the thing. Whether it is scientific or unscientific, when you are in a problem, how you are solving the problem, how you are seeing the problem, that is the questions it is put in front of the mankind. You are absolutely right, Dr. Gopalakrishnan. I completely agree with you. That's why I put in my title, COVID, that particular things to hear.
One last question. There were lots of questions, but then I will restrict myself to one last question. Over-the-counter antibiotics.
Yes.
I don't see it working in India because we don't have regulation. We have rules, but there is no implementation of these rules. We see that with the traffic rules, no implementation of the traffic rules, and I don't. The medical over-the-counter, the pharmacy people, they become doctors basically in most places, and it's very, very strange and very horrible because more than antibiotics, painkillers are dispensed without any restriction, and we know that all these painkillers affect the kidneys.
Yeah, absolutely correct, Dr. Gundusikapan. When we have an international meeting, this point was discussed. And what I want to come to know that many medical shops has not been audited for years or decades, or all those things are going on. At present, last state is Telangana, who has put on words that without showing doctor, you can't take antibiotic, but it in words. But at least in Kerala, what are the results coming? Though there are others, concern has to be taken care of. And government is sending 2 people to the everywhere for the awareness and telling not to take antibiotic without doctor prescription. So here the question is, our everything is pushed to the wall. So we need to, if government doesn't do it, we need to be aware, we need to be educated, we have to know these logics, and we need to come forward for the movement. And what I'm requesting also, last part of my talk, just do this kind of a things without showing, no doctor, don't antibiotic, and things your antibiotic consumption of your family. Are you getting reduced and you're healthy or not? Just a simple test. That would run. And Jabai, I just want to tell you, Professor Jha is here. Our Vikas Bansal and Sadhana in Madhya Pradesh, they arranged a talk with the hospital, around 300 doctors, and Dr. Giriraj was there. And we discussed this point with all doctors, how they treat all those things. So this awareness has to come. Only Breakthrough Science Society alone can't do it, and it can't be done with the help of a doctor. And let's see, do the awareness and education to stop these menace. So I completely agree with you. Rules has to be very, very particular to do that. Otherwise, a very danger in front of the mankind.
Thank you, Parimal.
Yeah, thank you. Thank you.
Okay, next question.
Good evening, sir.
Good evening.
Sir, bacteriophage virus are there, not to kill the bacteria. Instead of antibiotics and antibacterial, can we produce such viruses that can kill all types of bacteria?
Certain papers can, that is called bacteriophage, which is isolated from the soil, and this is kind of a custom-made things, but it has not been that much successful. It is still under trial. So this is one of the novel idea scientists are doing it. I agree with you.
Thank you.
I have a small question.
Yeah,
Dr. You were talking about a new method, approach for crossing the MRSA resistance, the methicillin resistance. In 2024, you said that they were able to cut and paste a few sequences to overcome the resistance. Can you slightly elaborate on it, if possible?
Actually, when a bacteria giving the resistance genes to offspring, there is a biological process called recombinase. Recombinase is a DNA binding enzyme. Part of the DNA it cuts, which is resistance, and it will be inserted, paste to its progeny. So that's called recombination. It's a human also, but bacteria does it. So if you don't allow that particular enzyme to bind to the DNA, which contains the resistant gene, then it will not be cut, or even be cut. If you don't allow it to be pasted, then this information of resistance can't go. Can you discover any drug which is allow not this protein to bind to the DNA, or if it's there, it will not do the ligation or pasting? That is the new resistance, what a new technique or new novel thought. It came in the science advance. So that's why I just told you, Dr. Ramachandran. Am I clear?
Thank you.
Hello.
Yes, come forward. Next question.
Looks like there are not many others asking questions. And so if there is time, then I can ask more questions.
Okay. No, there is a question in comment box.
Okay.
Parimal, can you see the comment box?
No, you have to read.
Okay.
Okay,
So,
okay. Let me see this. Oh, you can. What is that? Can other
How
people
can antimicrobial use
in veterinary
in
and animal husbandry be accessed and addressed to the influence and behavioral change among students and future? Yeah. So I have given a list of antibiotic, OTC, streptomycin, how they are getting used, and there is a strict measure coming now from the government, and there is a checking coming, or it should be coming. When you grow animal farming or there, this kind of antibiotic uses is no more. So those selling and all those restrictions coming, but there is a rule. It is on just on papers. So this is coming, and it is a very dangerous. As I told, 75% antibiotic used in this animal farming and veterinary things. So it has to be very, very careful. And strict rule has come. Like that, also second generation and third generation antibiotic case of cases and all. But it has to be executed, and we have to build up a movement to safeguard ourselves. So this is one of the things what I just suggesting here.
Any more question?
Good evening, Parimal. This is Tabrez.
Yes, Dr. Khan.
Yes, this news in newspapers recently and even from since long back, there is an argument going on like too much cleanliness will actually harm your children, this kind of thing, which will not improve their immunity or something like that. So, how should we balance this argument?
Well, Dr. Khan, this has been discussed also, whether vaccine is good or COVID or not. You can always raise these questions. So what we are going to tell here, we are not restrict that every second you will not be exposed to these things. But what we are telling, restricted you, all those kind of things has to be done. Immunity raising and all those things and bacterial infection through that washing hand doesn't go side by side. It's just kind of what I can tell you. You can feel what is happening and what needs to be done and what needs to be allowed or not. That's what I'm telling. Nobody's telling it's a sterile environment and you keep your baby in a kind of a system in hospital, don't come out for that. No, that's nobody's telling. But the telling thing is certain part of the things. I just want to tell, when you take food, do you wash your hand or not? We do, all we do. After certain things, we wash our hands, right? We do that. So that needs to be restricted, especially from the coming from the hospital, doing for all those. So this is nothing related to immunity growth and all those kind of, this is a misconception going on. There's too much to read about all those things, what is doing it. And see that Pennsylvania, how they have done it. Okay. Every day the kids doesn't catch cold, right? It's not for all these immunity things. It's cold doesn't mean it is temperature low. It is an organism which gets it infected, right? It's a misconception that I have cold. Cold means nothing. You have infected with the organism. How severe it is, that doesn't matter. Whether it requires antibiotic or not, that is doesn't. But you got infected. Otherwise, why you will get this kind of sneezing and all. So this is the concept. What I just want to tell you.
Okay.
Any more question?
Yeah, I think Dr. Gopalakrishnan, you have some further questions? Or?
No, yeah. So a lot of things that we need to discuss in Breakthrough Science Society. For example, you touched upon climate change, global warming, and how some old bugs are getting, are surfacing, and they can be devastated for
Yeah,
us.
this is one part of that from the climate change side effect, contributing antimicrobial resistance. So, because
And
doubling time also increasing, that's true.
and and the thing is that we are signing petitions against war, but we are not able to stop the wars.
Yeah, so this is
There
another.
is no there is no what do you call conscious willing if effort from the international community. The international community has collapsed. There is no international order. The global warming is going to increase, and we will have more extreme climate events, and then the kind of things that you mentioned about old bugs surfacing and the new bugs getting more and more resistant, and they will be spreading more parts of the world to the more to even to the temperate climate regions. They are becoming warm, and so these the tropical bugs will also go to the temperate zones. So where do we stop? When what can we do? Just washing hands is not enough.
Yeah, absolutely. That's why I told you, we have to build up a strong movement for OTC of antibiotic uses. You need to have a new antibiotic. More research and development has to be done. We need to take measures in this ICU practice of the hospital, and all this waste product which is coming out of hospital, whether this has been properly sterilized or not. Show this in this other industrial things and all. So what I just want to tell you, it is a 360-degree feedback. What is the way it is? Once is related to the entire things. It's not isolated things what's going to happen. So it's a, but one thing you are right, and there, this is a war when happen, bombing happens, everybody can know is getting destroyed. But this is a silent killer. And now here, Professor Mukhopadhyay there, who has given, I have forwarded to several lectures given on climate change by Professor Mukhopadhyay, our Breakthrough Science Society president. He's also touched upon several climate things on what are the dangers looking. One of the side effects I'm telling, but what are your last point telling is very, very important. When war happens, bombing happens, we can see, I witness this happened. But when it is slow killing happens through this antimicrobial resistance, we don't care until it comes to the nodal point. So here is the awareness, here is these things what you are telling. We need to build up this movement. If you are all logically, scientifically convinced, this slow change evolution in hamper when change is nodal point and create a big like pandemic like COVID. And I'm telling you, all the scientific international forum, G8 or whatever there are a lot of things are coming to antimicrobial resistance, lot of circulation coming. But when going to the ground level, still a village lady has 10 to the power 14 MRSA infection for her, this what is called urinary tract infection, because they can't use tissue paper, especially for the female ladies. All your parents and you are telling male and female the difference here. Getting infected is very difficult. This is one of the way. That's why I just want to appeal to all of you.
So what you are saying, describing, is that we need more ASHA workers. We need more primary health.
Absolutely. Actually, this is the thing. What happens in Kerala? Those, I think Dr. George is here. Though they are sending people to the different houses to get the awareness, but when coming to the ASHA workers things and all, it is not any that much of things from what is called government level. So it is other way around. So it is whether we want to lead or our next generation, all whole world wants to lead a healthy life. It is in our hand, and we only know one thing: scientific movement, collective scientific movement to stop this menace. And
I mentioned ASHA workers because ASHA workers can help in this education.
Absolutely. Contrary for the ASHA workers there, they're fighting for their salary minimum things, and their work they are doing it is a completely quite opposite, which give us all scientific worker like you and us very much pain. And we need to do it's not a one way only scientists or doctors. It's the entire society has to come. Do we want to have another COVID or not? That's the question. Question mark I put it. I'm not told you what about these things. So this is the things I'm doing, what we are appealing to everybody. Let us practice ourselves first. If you have here 50 people, we will go 100, 100, 200. Like that, we can do that. So let us do at least from these things. But this washing hand is not the solution, Dr. Bush, what I'm telling. Just a prevention.
No, it's important.
That's what I'm just telling, because this is very easy because COVID has shown us.
Washing hands and legs.
Parimal,
yes.
There is a question in comment box. Can you address it?
What is that question? Regarding the climate change thing, just curious, is it possible to genetically engineer plants to make them grow faster, like merging the DNA of bacteria which produced very fast and using this feature to multiply? So, Dr. Kishan Das, what you asking questions, you need to understand that bacterial genome and plant genome. So you are asking for the bacterial genome and plant genome fusion to do something, to bacteria produce very fast and using this feature to cell of a plant, thereby helping the plant to grow faster. You are coming into genetically modified plant, what has done not to infect those kind of things. If I understand correctly, that have several other debates. But that knowledge, what we have used to use the genetically modified plant, not for this production, but not to infect, you asking that same knowledge can be utilized for development of some medicine or something to here. That's a different questions. Two completely different things. So I dealt with the three parts. You are asking the fusion of a plant gene with a bacterial gene and to get something yet to be seen. Second thing, the way the information is used to protect plant to getting infected from that environment bacteria is one knowledge. With a similar kind of a knowledge could be imparted to the human to raise drug. That's a different question. So that's what I understood from you. If you're not clear, you can ask me further, Dr. Das.
Okay, so now it is the time to stop. Still one or two questions we can address. Because we are running out of time now.
Yeah.
Okay. So we again thank you all, and thank you, Dr. Misra, for his wonderful talk. Thank you.
Thank you. Thank you. Thank you. Thank you all.